work this so that when you saw your grade, it was actually reflective of your grade, and it's not, because Canvas is stupid. So I did go through, those of you who did the extra credit, okay, you were provided with those points. But the way Canvas works is say take a look at that as an individual assignment and provides and calculates the grade based on that, which again is stupid. So to quickly determine what your grade is simply add up all of your points and then divide it by 200, and boom, you have your percentage. You can multiply that by 100 if you need to figure out the total numbers. But you do that and you can determine what your grade is at this point. So it's all the points that you've earned, including the extra credit ones, and then just divided by the 200, which is the total number of points that are available at this point in time. So, and again, Canvas. So, but, okay, any other questions or concerns? Okay, awesome. So, Jeffrey, I'm gonna come on up. And then after Jeffrey's presentation, Gabe will go over his article. So, and then to use this, the pointer is up top here. And then to go forward, simply that, backwards, and don't hit this button because it'll give you a blank screen, okay? Okay, there you go, Jeffrey. And speak loudly so everybody can hear you. And I'm going to say that to everybody. Looking at you, Sarah. Okay.
All right, good morning. My name is Jeffrey, and my NSF proposal is going to be about microplastic studies in the Big River ecosystem. So plastic is a ubiquitous environmental pollutant. This plastic is found in several ecosystems. Like you can see, these are like this is the ocean, and then this is like a landfill, right? So we have plastics in the rivers, in the water bodies, and then in the land. Now, between 1950 and 2017, we produced about 9.2 billion tons of plastic, and we've only incinerated about 1 billion tons of this plastic that we produced, and about 5.3 billion has been discarded. So where do we discard all this? We discard this in the river bodies, and in the water bodies, and in the oceans, and then in the landfills. But then experts say by 2050 there will be more plastic in the oceans than fish, right? Because if you continue depositing these plastics in these oceans, then it means that these fish and these marine organisms will be feeding on these plastics. Now, we can categorize plastics into different types based on the size. So if you have a plastic less than 5 millimeters, then we can say this plastic is a microplastic. So microplastic generally bioaccumulate in the environment. So you can see humans are the biggest culprits here. So we litter plastic all over in the environment. In the industries, they keep producing more plastics. Now, these plastics can biodegrade in the environment by UV and mechanical stress. We produce more microplastics, and these microplastics are again ingested by these marine organisms like fish, etc. And then, of course, these bears also feed on these fishes in these water bodies. So we keep transferring the microplastics, we put them in the ocean, the fish is degraded, and then these higher trophic organisms also keep feeding on it. Now, it has been estimated that humans eat over 40 pounds of microplastics in our entire lifetime. You might not see this, but several research has shown this. Now, those of you who like seaweeds, now it has been found that most seaweeds are usually sequestered with microplastics. So the more of these food that you're eating, you probably are also consuming more plastics. Now, Blanc Mittal conducted a study, and then they found out that inhaled micro and nanoplastic particles may pose higher risk of cancer in the future. And microplastics can also serve as vectors for pathogen transmission. Now, when we say vectors for pathogen transmission, we mean microorganisms and other dangerous chemicals can be sequestered on these microplastics, and then as these microplastics are being transferred to the trophic levels, these microplastics are made available to us, and then if there are any like pathogens, we can also get them. So this term plastisphere is like a general term used to describe like a diverse group of microorganisms that are living on a plastic surface. So when we have biofilm formation, or when we have like other groups of microbes living on the surface of plastics, we call this a plastisphere. So this is like a general overview of what a plastisphere is, like a microplastic interaction. So here we can see the microplastic fragment surface, right? And then we have biofilm formation. This is a Vibrio species biofilm, and then these are like a multi-species biofilm. And then we can see different types of microplastics here. We have microplastic fiber, we have microbeads. Now, you can see microplastics, you can see biofilm formation on the surfaces of these marine organisms as well, right? And then you can see like heavy metal accumulation on the surface of the fragments of the microplastic, like aluminum, you have copper, and then we have zinc. And then of course, we can also see this chemical here called DDT. This is a very toxic chemical which was probably used back in the day for fishing, but it has been banned by the EPA. But then these toxic chemicals can also be sequestered on the surfaces of these plastics. So you can see if this microplastic gets into your food or in your water, you are probably being exposed to all these interactions. So I chose this site for the NSF proposal because it's a very contaminated area. So back in the 1700s, there are a lot of like mines here, and these mines are like lead mines, right? So they used to mine lead in this area. And then these lead mine drainage drain from here into this big river here. So this big river contains about 8 million tons of lead, right? And it's you can see here, this is Southwest Missouri Superfund National Priority List. So this is a very huge problem being addressed by the Environmental Protection Agency. Now, the economic cost of antibiotic resistance to the US economy alone is an estimated 20 billion in direct health. And then many heavy metals augment proliferation of antibiotic resistance due to their antimicrobial properties, because metals in itself can serve as antimicrobial particles. So if you have an ecosystem where you have a lot of metals, there's a higher chance that these microbes may also develop other resistances. Antimicrobial and heavy metal tolerance in microbial communities is facilitated by the plastisphere formation. So we saw all the interactions on the plastisphere, and we saw how these interactions can affect us. So my research objectives. Now, the first one, I want to determine the dynamic processes of biofilm formation on different microplastics submerged at varying depths and times in the big river. And I also want to determine the influences of biofilm formation on the surface morphology, physicochemical properties, and microbial community profiles on microplastics in the big river. My research hypothesis. I hypothesize that biofilm formation on microplastics may increase with increasing depth and exposure time. And the biofilm reduces surface hydrophobicity of microplastics at varying degrees. And dominant microbial species might change with increasing exposure time and depth, as well as the type of microplastic. And then more than 70% of microbial isolates from Big River will carry both antibiotic and heavy metal resistant genes. So this is like an overview of my experimental design. We're going to use these 5 classes of microplastics. So we will acquire microplastics polyamide microplastic, polyethylene terephthalate, polypropylene, and polyvinyl chloride. So we expose these microplastics at these depths, so 2, 6, and 12 meters. Now, Tu et al. Published a paper similar. They did something similar, but they used PE microplastic, right? And then this is kind of a stainless steel cage, which is filled with the microplastics, and then they kind of use some varying weights. They put some weight at the bottom of the cage so that the whole thing will just like suspend, right, at different depths. And then the exposure time will be for 30 days, 60, and then 90 days. So we put this in the big river, and then we leave it for 30, 60, and 90 days. So we will take our samples on these days. Now the size of the microplastic will be 4 millimeters, and the density should be approximately 1 gram per cm. Yeah. So once we collect our microplastics in the big river, we bring it to the lab, and then we perform these microbial analysis. Now, for this part, we need to work on both the microplastic and the biofilm. So what you see here, these things are the biofilm on the surface of the microplastic, right? So we analyze the microplastic alone, and then we analyze the biofilm alone. Now, the microplastics here, we need to undergo what we call the FTIR to see if there are any functional chemical group changes on the microplastic surface. So we take the virgin microplastic and then we compare it to the one which has been exposed for 30, 60, or 90 days. And then we check the surface hydrophobicity. So usually more plastics are hydrophobic, right? But then when you expose these plastics to a new environment where these microbes are kind of interacting with these microplastics, then we assume that the hydrophobicity of the microplastic might change. So we need to determine the change in surface hydrophobicity compared to the virgin microplastic. Okay. So basically we are done with this part right now. So we come to analyzing the biofilm. So we can extract community DNA from this microplastic, from these biofilms, and then we target the 16S rRNA gene, the V4 hypervariable region, using these forward and reverse primers. So once we're able to sequence the community DNA, we can find once we know the sequences, we can either use further analysis like maybe bioinformatics to see if we can find some sequence homologies in terms of what genes might be present in this community DNA samples. Now, we can also do an extra test where we kind of form something like a selective media where we use some like common antibiotics like ampicillin, vancomycin, tetracycline at 100 microgram per ml. And then we also might add certain metals. We know that the big river is heavily polluted with lead, so we know lead will be present. But we can also add other metals from the big river, because I mean we assume lead is there, right? But then we also assume other metals can also be present. So we will undergo this EDX analysis. This is energy dispersive X-ray analysis. Yeah. So this quantifies the elements on the surface of the biofilm, right? So if there are other metals, like other heavy metals, this will give us these metals, and then we can add these metals to our culture, such that every microbe or every microbial species that can be resistant to both these antibiotics and these heavy metals, we can further select them as a resistant species, and then we do further analysis on them. Okay. Now we can also determine the total biomass, the total biofilm biomass by using this crystal violet stain, and then we measure the absorbance at 5, 9, nanometers. This will quantify the total biofilm on each microplastic, because we believe that some microplastics might be more ideal for microbial colonization than the others. And then we can also quantify live cells, dead cells, and the extracellular polysaccharide secretions, the EPS sheets that might be formed on the surface of the biofilm. So we use these three different dyes. This SYT09 will stain live cells in green, and the propylium iodide will stain dead cells in red, and concanavalin A will stain the extracellular polysaccharide sheets. So expected results. Biofilm biomass on microplastics at increasing depth and exposure time will be significantly different due to changes in temperature, light intensity, organic matter, nutrients, as well as other environmental factors like dissolved oxygen, pH, etc. Surface hydrophobicity of MPs is expected to vary significantly due to the unique chemical composition. But the extent of chemical change, we don't know, will depend on the microbial community present. The dominant microbial species will vary with microplastic type. So we believe that maybe microbes that will be dominant microbial species on PE versus PET might be different. And also the exposure time and the depth of the exposure will also influence the type of microbial species present. And then due to the high lead concentration in the Big River, we also expect that more than 70% of these microbial isolates might carry both antibiotic and heavy metal resistant genes. So in conclusion, microplastics provide ideal surface area for biofilm formation, resulting in proliferation of diverse microbial communities. And we said these microbial communities share kind of information in terms of genes, right? Through horizontal gene transfer. Understanding the interactions between global microbes and microplastics in contaminated ecosystems like Big River will provide more insight into developing alternative solutions to tackle emerging resistance chains. Thank you.
Okay. See, questions for Jeffrey?
Can you go to slide 10 real quick?
Slide 10.
Yeah, the next one. Where does the greater than 70% of microbial isolates carrying antibiotic and heavy metal resistant genes, where does that 70% come from?
So we said that the Big River ecosystem is mainly polluted with lead, right? Right. So now, because it's mainly polluted with lead, the assumption is that microbes may carry other genes, right? So these genes may be antibiotic resistance genes, right? So it's like the metals itself may serve as like antimicrobials, right? So the organism might find a way to like pump these metals out of the cell, right? So if you have, once they are pumping these metals out of the cell, they also might pump out like maybe antibiotics, right? So the most organisms that are resistant to heavy metals are also resistant to antibiotics. So if we have more organisms resistant to lead, then there might be other organisms, there might be more organisms which might be also resistant to antibiotics. So this is just something that I'm expecting, but it's also an experiment. So it might be that after the experiment, I might not see this.
Yeah, I didn't know if like other experiments had been done similar to this that like had a quantified value.
Well, I wouldn't say there's a quantified value, but this is just something that I think might happen.
Okay, thank you.
From a river mechanics standpoint, where are you putting your sampling devices? Because if you put it in something like an eddy where you have a circulation of water where there's like, it kind of sits there, or if you have it in a high flow environment in the thaw wag, or if you have it in a low flow environment, that could affect the different exposure limits of lead or other, like, or microplastics even, because you might have some areas that are low flow where the river kind of circulates the same water a little bit more, so then you'll have, like a greater chance and concentration of what could be flowing down the river in terms of like, microbes or lead, but then if you have it in a really high flow environment, it might be going by too quickly for it to actually like affect your, experiments. Like where in the river are you placing these? And I kinda understand the depth part.
Yeah, I didn't really think about this like a question in terms of where, like the exact part of the river, but I think we don't need like a high tide area. We don't need where like the river is like moving like so fast. Like maybe I'll choose a site where the river is like probably like stable. Like I'm talking about the current of the river is not that strong, probably. But I'll do more reading on this and see. Yes.
For your experimental design, why would you do it like immediately in the river first? Like, would it be better to do it like collapsing to like control for like temperature and light exposure, keep those constant to really see what effects?
Yeah, so like most researchers have done, most research on microplastics are usually done in the lab, right? Lab setting. But this is one of the challenges in this area of research because it's like you want to simulate what is happening in the river bodies in the lab, but sometimes the simulation is not that accurate. So it's like you don't get environmentally realistic concentrations, right? So whatever you're trying to simulate might not be the exact thing in the environment. That's why we want to incubate this in situ in the environment to see the actual effect.
Anyone else has questions?
So are you exposing, so you're gonna have the exposure depths at 2, 6, and 12. Are you going to have each of those be exposed, like have like 3 sets of those at 30, 60, and then 90 days as well?
Yeah.
So you'll get all of the depths and all of like the exposure time, like, combinations?
Yeah.
Awesome.
Yeah. And we're gonna replicate this about 3 to 4 times. So for each microplastic, we have like several of them. So this cage that you see here contains more than 200 pieces of this for each of the microplastics, because I mean you're just cutting the microplastics into this size and just putting them in, right? So for each sample, so if you take this cage will contain 200 microplastics. So you can get a lot of samples to do more analysis.
Just had like a quick thought. That's obviously experimental, theoretical, but like maybe what would be the end like way of keeping extra microplastics from being introduced into the ecosystem? Like, do you have a way to just try and limit the amount that just gets swept away by the river?
No, honestly, I don't. Now,
Yeah.
this is just like trying to maybe find emerging resistant strains in this polluted environment, right? But I don't think anybody has like a lasting solution of how to
Yeah,
use.
I was just like curious, like just to make sure you don't get any microplastics spread anymore.
So like for the 30, 60, 90 days, is there like a season that you're hoping to do this in, or are you hoping to do it in like multiple different seasons? Because in the winter time, you're gonna have lower nutrient levels anyway and lower temperatures, and it's a drastic difference, especially in Missouri.
Yeah, I also didn't think about that, so I'll incorporate that in my write-up.
See, I also have a couple of questions too. And actually, thank you, that was one of my questions. So, but I guess, first of all is, besides the heavy metals and all, what sort of impact is there on plastics in this particular environment? Is it known to have a lot of other anthropogenic input besides
You mean
the lead
the-
mining, the big river? So is it just because of the influence of the heavy metals, or because, another way of looking at this too is, what has been the impact of microplastics on the microorganisms in this environment?
Yeah, so actually, I don't think any other work has been done on in terms of like what microplastics might be present there. I chose this site because I felt like microplastics are everywhere, right? Microplastics are polluted in all our water bodies, right? So if you have a heavily polluted ecosystem in combination with these microplastics, it might be very like detrimental, right? So that's why I kind of chose this to see if the microorganisms in this environment, how they will interact with these
So
microplastics.
you might want to include something about what are the overall anthropogenic impacts on this environment, and just have sort of a background besides the metal influence. And then, what sort of environmental parameters do you have for this particular river? Because one of the things that I was thinking of, along with what Josh was mentioning earlier, is where exactly you'll place these devices to be exposed, and like you figure, I don't know anything about this river, so what is the depth of the river and how much, and then following up about the seasons and things like that, you really do have to provide a good understanding of the different environmental parameters. I mean, because when I'm thinking about 12 meters, that's going to be like 36 feet. Is the river even that deep?
The river is more than 100 meters deep. Yeah, the river is more than 100 meters.
Okay, so you might want to actually, in your report, provide sort of a map of the river itself and some of these parameters. So, yeah, Josh.
Something also to consider is the proximity to historic lead mines that you want to put this in, because lead is so dense that even with like currents and things like that, like we find a lot of lead in the environment, especially in aquatic environments, being mixed in with the sediments. Since lead is relatively aqueous, it won't really just float through the water and hang out a lot of times. So what'll end up happening is, like, if you're really close to a historic lead mine, there might be some more mixed in at different stratigraphic levels within the river. But then as you go further down, you'll see more, like it'll start decreasing in value more and more. And there's gonna be kind of these sweet spots where there's gonna be heavy concentrations of lead, because that's the time it takes to settle down into the sediments, or like get washed up kind of in the bank sediments and everything like that, just because it's so dense compared to the water and everything else around it.
And then, my other question is, how are you going to separate the biofilms, the microorganisms, from the microplastics that you can do, like the culturing and the DNA extraction that you're proposing to do?
Yeah, so usually with analysis of the microplastic, I think that is quite straightforward, right? We just take off the biofilm.
Okay, how do you do that? Are you going to scrape it, or are you going to use a chemical means, or?
No, we're not gonna use we're gonna scrape off the biofilm, but we're gonna.
Okay, so then that raises the question, how big is your microplastics?
The microplastics are, you mean like the size? They will be maybe 5 by 5 squares. Yeah.
Okay, so you'll need to think about this, okay? But, yeah, and I don't know if physically scraping it will work. So what you need to do is actually think about the individual steps. And so, as we talked about, towards the beginning of the semester, just preparing samples and thinking about the Okay, don't take all the questions as a bad indicator or whatever. We just wanna know more, and it's a very good topic, so, okay, thank you, Jeffrey.